lapel.
Welcome, curious minds, to another deep dive.
Hello.
Today we're tackling a topic that's absolutely fundamental to pharmaceutical sciences. Cytochrome P450 inhibition, specifically in drug discovery and development.
That's right, a really core concept.
And we know some of you, maybe like our listener today, might be gearing up for a PhD qualifying exam in this very area. So think of this as your express lane to understanding the why, what, and how of drug metabolism and those crucial drug interactions.
Indeed. We're going to unpack a pretty comprehensive chapter on this, aiming to distill the, let's say, complex science into clear, repeatable concepts.
Exactly.
Our goal here is to really arm you with a solid understanding, from the kinetic models you see in the lab right through to their real-world implications for patient safety. Essential stuff for those exams.
Absolutely. We'll summarize the key points, break down the big concepts and pathways into hopefully simple language. Explain it in a way you could feel confident repeating in a lecture setting.
And maybe throw in some real-world drug examples where we can.
Yeah, where the material allows. Okay, let's get started. IR, the critical role of CYP inhibition in drug development. So, to kick us off, why is this topic, CYP inhibition, why is it such a big deal? What makes studying how drugs interact with these particular enzymes so important in the pharma world?
Well, it really boils down to two things. Safety and efficacy. Always.
Okay.
Our bodies have this incredible internal cleanup crew of enzymes, mostly in the liver, the hepatic cytochromes, P450 or CYP.
The DMEs, drug metabolizing enzymes.
Exactly. And these DMEs, they are responsible for breaking down just a vast number of drugs. So if one drug gets in the way of another drug's breakdown, yeah, you can get into trouble. It can lead to dangerously high levels of that second drug in the body, causing toxicity, or the opposite problem gets broken down too fast, becomes ineffective.
So a metabolism-based drug interaction basically means one drug is changing how fast another one gets cleared out.
That's the essence of it.
Which could make a drug either too strong, causing side effects, or maybe too weak, so it just doesn't do its job.
Precisely. And here's the kicker. The majority, really the vast majority, of clinically significant drug interactions, or DDIs, they happen right there at the level of these CYPs.
Wow, okay.
Now, clinical DDI studies, testing these in people, are incredibly expensive, and they take a long time. So there's this huge push in the industry to screen for these interactions much earlier.
Using lab tests, the in vitro stuff?
Exactly, in vitro systems. This gives critical information way earlier, for finding leads, for optimizing drug candidates, and just making smarter decisions about which drugs move forward into those expensive clinical trials.
Saves time, saves money, and hopefully prevents problems later.
Absolutely. It's foundational.
Okay, so let's talk specifics. The source material highlights 6 main human hepatic CYP isoforms. The big players in drug metabolism. Can you list those for us?
Sure. These are the ones you absolutely need to know. CYP1A2, 2C8, 2C9, 2C19, 2D6, and the big one, CYP3A4.
3A4. Always hear about that one.
Yeah, it handles a huge percentage of drugs. So these 6, they're the workhorses. The ones most commonly checked when evaluating new chemical entities, NCEs, as potential inhibitors. 2, the how. Measuring CYP inhibition in vitro.
Okay, so if we need to predict these interactions early on, how do scientists actually, measure CYP inhibition in the lab? What's the basic approach?
The common strategy is to monitor how a test compound affects the metabolism of a known CYP probe substrate.
A probe substrate, like a specific customer for that enzyme.
Exactly. A known client for a specific CYP enzyme. We use human liver microsomes, HLM usually. Think of HLM as little packets containing these enzymes taken from human liver tissue.
Got it. So first you see how much product that specific CYP makes from its probe substrate under normal conditions. That's your baseline activity. Then you add your test compound, the potential inhibitor, and see if that production slows down. Makes sense. And generally these reactions follow Michaelis-Menten kinetics, that classic enzyme curve.
Yes, typically they do, which is great because it allows us to derive key constants. Km and Vmax.
Right, Km and Vmax. Can you just quickly remind us what those represent for someone prepping for an exam?
Absolutely. So Km, the Michaelis constant, it's the substrate concentration where the reaction runs at half its maximum speed. It gives you a feel for the enzyme's affinity for its substrate lower. Km means tighter binding, generally.
Okay.
And Vmax is simply the maximum rate, the top speed the enzyme can work at when it's completely saturated with substrate.
Got it. Affinity and top speed.
Right. And if an inhibitor is present, we can then figure out values like K for reversible inhibition or Ki and kinact for irreversible inhibition. These kinetic parameters are really the core data we use to try and predict what might happen in a whole person.
Crucial pieces of the puzzle. Now, the source material talks about how these lab assays have evolved. There were older methods.
Oh, yes. There's been a definite progression. They mentioned fluorescence-based assays first.
Those sound like they'd be fast for screening lots of compounds. High throughput.
They can be fast, yeah. But the source points out some significant drawbacks, like interference from the test compounds themselves messing with the fluorescence signal.
Like background noise.
Exactly. Or sometimes they lack specificity, meaning you might need to use individually expressed CYP enzymes made in a lab rather than the more, let's say, physiologically relevant mix you get in human liver microsomes.
So maybe not the most realistic picture.
Could be. And then there were radioisotope-based assays, better selectivity, often better substrate solubility.
Sounds like there's a but.
But they usually require a separation step before you can analyze the results, like chromatography, and that really limits how many samples you can run quickly. Not great for high-throughput screening.
Okay, so fluorescence has interference issues, radioisotopes are slow. What's the current gold standard that gets around these problems?
That would definitely be the LC-MS-MS method, liquid chromatography tandem mass spectrometry.
LC-MS-MS, hear that a lot.
You do, and for good reason. It offers really superior throughput compared to the older methods. It's flexible. You can use various probe substrates, different enzyme sources like recombinant CYPs or HLM.
Versatile.
Very. And the instrument itself is highly selective, very sensitive, and fast. It lets you do rapid gradient LC separations and, crucially, detect multiple metabolites simultaneously. Super efficient.
Like being able to track several different products at once.
Precisely. For instance, it can easily separate and quantify different hydroxylated forms of testosterone that are produced by different CYPs and HLM, all in one go.
Okay, yeah, that efficiency makes sense for screening thousands of potential drug candidates. It's about getting clean, precise data and getting it fast to make those early decisions.
That's the name of the game. The source even gets into the details, like the specific equipment robotics, like Tekken for handling liquids, the triple quadruple mass specs for analysis. It outlines how you prepare the HLM, the cofactor NADPH, how you optimize the reactions to make sure they're linear, you're not using up too much substrate.
All the practical details you'd need to actually run the experiment.
Right. And it shows specific LC-MS-MS analyses for individual CYP marker substrates tracking, say, acetaminophen formation from phenacetin for CYP1A2 or 4-hydroxydiclofenac from diclofenac for CYP2C9. Seeing those specific molecular changes tells you exactly what the enzyme is doing and how the inhibitor is affecting it. 3, unpacking the what types of CYP inhibition.
Right, so we've run our fancy LC-MS assays, we've got our data.
Yeah.
Now, what does it actually tell us about the kind of inhibition happening? The material makes a clear split between two major types, reversible and irreversible inhibition.
Yeah.
Dig into reversible first.
Okay, so with reversible inhibition, the inhibitor binds to the enzyme, but, and this is key, it can also come off again, it dissociates.
So the effect is temporary.
Temporary, and it depends directly on how much inhibitor is around. The general kinetic model describes this equilibrium where you can have the enzyme bound to substrate, ES, the enzyme bound to inhibitor, EI, and sometimes even a three-way complex, enzyme-substrate inhibitor, ESI. The main point is that the presence of the inhibitor reduces the amount of active enzyme-substrate complex, the ES, and that slows down the rate of product formation.
And within reversible, there are different flavors, right? 4 main types mentioned.
That's right, each with a distinct way of interacting.
I always think of these like different ways someone could interrupt you.
That's a good way to think about it. First is competitive inhibition.
Okay, competitive. Sounds like what it is.
Exactly. The inhibitor directly competes with the substrate for the enzyme's active site. Like 2 people trying to sit in the same chair, only one fits at a time.
So if you add way more substrate, you might overcome the inhibitor.
You often can, yes. That's a hallmark you see in the kinetic plots.
Okay, what's next?
Non-competitive inhibition. Here, the inhibitor binds somewhere else on the enzyme, not the active site. And it doesn't care if the substrate is already bound or not.
So it binds regardless.
Right. And by binding elsewhere, it changes the enzyme's shape or somehow messes with its catalytic machinery, making it less efficient, like throwing a wrench in the gears, as the source mentions. The machine still turns, maybe, but slower, regardless of how much material you feed it.
Got it. Doesn't compete directly, just makes the enzyme worse overall.
Mm-hmm.
Third type,
Uncompetitive inhibition. This one's a bit more unusual. The inhibitor only binds to the enzyme after the substrate has already bound, so it only binds to the ES complex.
Only after the substrate is there. Huh.
Yeah. It essentially traps the substrate on the enzyme, preventing the product from being released or formed, like a jam in the machine that only happens once it's loaded.
Okay, interesting. And the last one.
Mixed type inhibition. As the name suggests, it's a mix. It has elements of both competitive and non-competitive binding. The inhibitor can bind to both the free enzyme, like competitive, and the enzyme substrate complex, like non-competitive or uncompetitive, often with different affinities.
So the most complicated one.
And probably the most common one you actually encounter with real drugs, because biological interactions are rarely perfectly one thing or the other.
Right. Okay, so for these reversible types, we get kinetic parameters like IC50 and Chi. What's the practical difference for someone trying to interpret this data?
Good question. IC50 is the inhibitor concentration that causes 50% inhibition. It's pretty straightforward to measure and useful for initial screening, like a quick rank ordering of how potent different inhibitors are.
A snapshot.
A snapshot, exactly. But Chi, the inhibitor dissociation constant, is generally considered a more fundamental measure. It reflects the actual binding affinity of the inhibitor for the enzyme. It's a thermodynamic constant, less dependent on the specific experimental conditions like substrate concentration.
So Chi is maybe more reliable for predictions.
Often, yes. The source notes that for simple competitive and uncompetitive inhibition, IC50 is roughly twice the Chi value if you run the experiment with the substrate concentration at its Kahneman value. For pure non-competitive inhibition, IC50 actually equals Chi.
But not for mixed types.
Correct. For mixed type, it's more complex, and you cannot simply assume IC50 is a direct estimate of Chi. You really need to do the full kinetic analysis, varying both substrate and inhibitor concentrations, run replicates typically 3, the source says, and use nonlinear curve-fitting software to properly determine the Chi value.
Makes sense. Get to the full picture. The source gives examples like fluvoxamine for CYP1A2, ketoconazole for CYP3A4, showing their IC50s. Those are known inhibitors used as controls.
Exactly. Well-characterized reference inhibitors.
Okay, now let's switch gears to the other major category, the one that often raises more serious concerns. Irreversible inhibition. The source calls this mechanism-based inhibition, or MBI. What's the crucial difference here?
The absolute core difference is that in MBI, the enzyme is permanently inactivated. It's not just blocked temporarily. It's either destroyed or chemically modified so it can never function again.
Permanently gone. Wow, how does that happen?
2 main ways described. First is suicide inactivation. This is where the enzyme itself starts processing the drug, but in doing so, it converts the drug into a highly reactive molecule, an intermediate.
That then attacks the enzyme.
Exactly. It attacks and forms a covalent bond, permanently inactivating the enzyme that created it. The enzyme basically commits suicide by processing the drug.
Grim. What's the other way?
The other is metabolite intermediate MI complex formation. Here, the enzyme metabolizes the drug, but produces a metabolite that binds extremely tightly, often covalently or quasi-irreversibly, to the heme iron atom right in the CYP's active site.
So it plugs up the catalytic center.
Right. The enzyme isn't necessarily destroyed, but its catalytic engine is completely jammed, rendered inert.
Okay, so either destroyed or totally jammed up. And the source lists clear signs that distinguish MBI from the reversible kind we just talked about. What should you look for?
Key characteristics for MBI. It's dose-dependent, obviously. It's pre-incubation time-dependent. The longer the enzyme is exposed to the inhibitor before you add the substrate, the more inactivation you see.
Because it takes time for the inactivation reaction to happen.
Exactly. It requires bioactivation. The enzyme has to actually try to metabolize the drug for the inactivation to occur. And this is a huge one for clinical relevance. Recovery of enzyme activity requires de novo protein synthesis.
The body has to make brand new enzymes.
Precisely. Because the old ones are gone or permanently broken. You can't just wait for the inhibitor to wash away, like with reversible inhibition. The body has to rebuild its supply of that enzyme.
Which means the effects could last much longer than the drug is even in the system.
Potentially, yes. The effects might be slow to appear, but can be much more profound and long-lasting. That's a critical point to understand.
Definitely. So how do we measure the kinetics for MBI? It sounds more complex.
It is a bit more complex. We use a kinetic model to define parameters like kinact, which is the maximum rate constant of inactivation, and Ki, note the uppercase I, which is the concentration of the inactivator that gives half the maximal rate of inactivation. It's conceptually similar to Kamiaba, but for the inactivation process.
Kinact and Ki.
Yeah.
Got it.
Another important term is the partitioning ratio. This tells you how efficient the inactivator is. It's the ratio of how many times the enzyme successfully processes the drug turnover versus how many times it leads to inactivation. A low partition ratio, say less than 10 according to the source, means it's a very efficient inactivator. The enzyme doesn't get many chances before it's knocked out.
High efficiency is bad news here.
Usually, yes.
Yeah.
The experiments involve pre-incubating the enzyme and inhibitor together for varying amounts of time before adding the probe substrate to measure remaining activity. This time course data lets you calculate an observed rate constant, COBS, and then use nonlinear regression or graphical methods like Lineweaver-Burke plots to determine the fundamental parameters KINACT and KI. 4. Predicting human drug interactions from in vitro data.
Okay, this is really where the rubber meets the road, right? Taking all this detailed kinetic data from the lab, KINACT, KI, and using it to predict actual drug interactions in people. That's the ultimate goal.
Absolutely. That's why all this work is done. To use the in vitro data to predict the DDI potential in humans before exposing patients, ideally. The key is to estimate how much the systemic exposure of a victim drug will change when the perpetrator drug, the inhibitor, is also present.
And we measure exposure using things like AUC, area under the curve.
Exactly. We often look at the ratio of the AUC of the victim drug with and without the inhibitor, or changes in its clearance rate, or its steady-state plasma concentration. These tell us how significantly the interaction impacts the drug's level in the body.
So for the simpler case, reversible CYP inhibition, how do we make that prediction leap from key to a potential clinical effect?
For reversible inhibition, we use mathematical models. These incorporate that crucial in vitro key value, but also other factors like the projected concentration of the inhibitor in the body, usually represented as I.
The amount of inhibitor actually there.
Right. And importantly, the unbound concentration, because only the unbound drug is typically free to interact with the enzyme. So we factor in Foo, the unbound fraction in plasma.
Okay. Keys, inhibitor concentration, unbound fraction. Anything else?
Yes, a really critical factor called FM. This is the fraction of the victim drug's metabolism that is catalyzed specifically by the CYP enzyme being inhibited.
So how dependent is the victim drug on that specific enzyme pathway?
Precisely. If a drug is almost entirely cleared by, say, CYP3A4, high FM for 3A4, then even moderate inhibition of 3A4 can have a huge impact on its levels. But if it's cleared by multiple enzymes, inhibiting just one might not matter as much.
That makes total sense. And the source gives a pretty handy rule of thumb for predicting competitive inhibition based on the Max key ratio. Sounds like a quick check.
It's a very useful initial assessment, widely used. You take the maximum expected concentration of the inhibitor in the plasma at steady state, Imax, and divide it by the chi.
Okay, Imax over key.
If that ratio is greater than 1,
DDI likely.
DDI is considered likely, yes, needs serious attention. If the ratio is between 0.1 and 1, a DDI is considered possible, worth investigating further.
And below 0.1.
Below 0.1, a DDI is generally considered remote or unlikely. It provides a quick, quantitative way to gauge the risk using the most conservative estimate of inhibitor concentration, Imax, for safety.
Very practical. Now, what about predicting DDIs from that trickier one, mechanism-based inhibition? Is that more complicated?
It is generally more complex, yes. MBI prediction models have to incorporate not just Ki and KINACT, but also the enzyme's natural degradation rate constant, usually called KDE or KE.
Because the body is constantly turning over enzymes naturally.
Exactly. The models need to account for the rate of enzyme loss due to the inhibitor and the rate of new enzyme synthesis and natural degradation. These models help estimate the change in the victim drug's AUC ratio, and they often need to consider both inhibition happening systemically in the liver and potentially inhibition happening in the gut wall for orally taken drugs, which can be significant for MBI.
So more moving parts to consider for MBI predictions. Given all this complexity, even with these models and rules of thumb, what are some of the big factors that can still make predicting these DDIs accurately, really challenging? Why is it not always straightforward?
Yeah, the source points out several critical factors that can muddy the waters. Big ones include choosing the right inhibitor concentration for the prediction. Should it be the peak concentration, Imax, the average concentration, IUG, maybe the concentration in the liver itself, which is hard to measure.
Right. What concentration is the enzyme actually seeing?
Exactly. Then there's the impact of drug binding, not just in plasma Foo, but also potentially binding within the liver microsomes used in the assay, which can affect the apparent chi value, especially for highly bound drugs.
Okay.
Accurately figuring out
on a pill.